तो सेक्शन 1 टेक्निक्स इन पैथोलॉजी आज हम लोग देखेंगे क्या ऑब्जेक्टिव्स। आफ्टर स्टडिंग दिस सेक्शन द स्टूडेंट शुड बी एबल टू द गेन knowledge and learn the following skills: learn the working of various parts of a light microscope, operate and maintain the light microscope, learn the broad principle of other form of microscope, discuss fixation of tissue for routine processing in histopathology laboratory. To be familiar with the equipment and basic technique of routine histopathology processing of tissue in laboratory, learn the routine H&E staining for tissue section, learn the technique, applications and stains used for frozen sections, learn broad principles and end results of common special histochemical stains. Section contains microscopy of various types, histopathology techniques and routine staining, frozen section and special stains. Microscopy of various types. Exercise 1. Light microscope are the type of microscopy. Recent advances in microscope. The microscope is the most commonly used piece of apparatus in the laboratory. It produces greatly enlarged image of minute objects. Common light microscope is described first, followed by other special types of microscopy techniques. Light microscope. A light microscope can be a simple or a compound microscope. Simple microscope. This is a simple hand magnifying lens. The magnification power of hand lens is from 2x to 200x. Compound microscope. This has a battery of lenses which are fitted in a complex instrument. One type of lens remains near the objective lens, and another type of lens near the observer's eyepiece. Lens. The lens piece and objective lenses have different magnification. The compound microscope can be monocular, having single eyepiece, or binocular, which has two eyepieces. The usual type of microscope used in clinical laboratory is called light microscope. A compound microscope has the following parts: a stand, body, optical system, light illumination system. A stand. This is a horse so shaped in monocular microscope. It gives stability to the microscope. Binocular microscopes have a variety of ergonomic shapes of a stand. This is monocular or this is binocular. Body. It consists of a limb which arises from the joint which with which microscope can be moved in comfortable position. The stand and the limb carry the following: first, body tube; second, a stage; third, knob for coarse and fine adjustment. Body tubes. There are two tubes: external tube which carries at its lower end a revolving nose piece having objective lenses of different magnification wheel. Internal tube is draw tube which carries at its upper end eye pieces. A stage. This is a metallic platform which accommodates glass slide having mounted object over it to be seen. A stage is attached to the limb just below the level of objective. It has an aperture in its center which permits the light to reach the object. Slides on the stage can be moved horizontally or vertically by two knobs attached to slide holder. Just below the stage is substage. Substage, which consists of condenser through which light is focused on the object. The substage can be moved up and down. The substage has an iris diaphragm, closing and opening of which controls the amount of light reaching the object. Knobs for coarse and fine adjustment. For coarse and fine adjustment, knobs are provided on either side of the body. Coarse adjustment has two bigger knobs, the movement of which moves the body tube with its lenses. Fine adjustment has two smaller knobs on either side of the body. The fine focus is graduated, and by each division, objective moves by 0.002 millimeter. Optical system is comprised by different lenses which are fitted into a microscope. It consists of eyepiece, objective, and condensers. Eyepiece: in monocular microscope there is one eyepiece, while binocular microscope has two. Eyepiece has two plano-convex lenses. Their magnification can be 5x, 10x, or 15x. Objective: these are made of a battery of lenses with a prism incorporated in them. Their magnification power is 4x, 10x, 40x, and 100x. Condenser: this is made up of two simple lenses and it condenses light onto the object. Light and illumination system: for daylight illumination a mirror is fitted which is plane on one side and concave on other side. Plane mirror is used in sunlight while concave in artificial light. Currently most of the microscope have inbuilt electrical illumination varying from 20 to 100 watt. Magnification and resolving power of light microscope. Magnification power of microscope is the degree of image enlargement. It depends upon the following: first, length of the optical tube; second, magnifying power of objective; or third, magnifying power of the eyepiece. With a fixed tube length of 160 mm in majority of a standard microscope, the magnification power of the microscope is obtained by the following: magnifying power of objective into magnifying power of eyepiece. Resolving power represents the capacity of the optical system to produce separate image of object very close to each other. Resolving power R is equal to 0.61 lambda upon N numerical aperture, where lambda is the wavelength of incident light and N is the numerical aperture of lenses. Resolving power of a standard light microscope is around 200 nanometer. How to use a light microscope? First, keep the microscope in comfortable position. Second, obtain appropriate illumination by adjusting the mirror or intensity of light. Third, when examining colorless objects, condenser should be at the lowest position and iris diaphragm closed or partially closed. When using oil immersion 100x, objective should dip in oil. Fifth, after using oil immersion, clean the lens of the objective with tissue paper or soft cloth. Other type of microscopy: dark ground illumination (DGI). This method is used for examination of unstained living microorganism, example Treponema pallidum. Principle क्या है? तो the microorganisms are illuminated by an oblique ray of light which does not pass through the microorganism. The condenser is blackened in the center and light passes through its periphery, illuminating the living microorganism on a glass slide. Polarizing microscope. This method is used for demonstration of birefringence, example amyloid, foreign body, hair, etc. Principle: the light is made plane polarized. Two discs made up of prism are placed in the path of light, one below the object known as polarizer and another place in the body tube which is known as analyzer. Polarizer shoots out ordinary light ray vibrating in all directions, allowing light wave of one direction or one orientation to pass through. The lower disc polarizer is rotated to make the light plane polarized. During rotation, when analyzer comes perpendicular to polarizer, all light rays are cancelled or extinguished. Birefringent objects rotate the light ray and therefore appears bright in a dark background. Fluorescent microscope. This method is used for demonstration of naturally occurring fluorescent material and other non-fluorescent substances or microorganisms after staining with some fluorescent dye, example Mycobacterium tuberculosis, amyloid, lipid, elastic fiber, etc. UV light is used for illumination. Principle क्या है? तो fluorescent microscopy depends upon illumination of a substance with a specific wavelength UV region, that is invisible region, which then emit light at a lower wavelength visible region. Electron microscope. This is used for studying of ultra structure detail of the tissues and cells. For electron microscopy, tissues is fixed in 4% glutaraldehyde at 4 degrees Celsius for 4 hours. Ultra thin micro sections with a thickness of 100 nanometer are cut with diamond knives. Principle क्या है? तो by using an electron beam of light, the resolving power of the microscope is increased to 50,000 to 1 lakh times, and very small structure can be visualized. In contrast to light microscopy, resolution of electron microscope is 0.2 nanometer or less. There are two types of electron microscopy: transmission electron microscopy (TEM) and second, scanning electron microscopy (SEM). Transmission electron microscopy (TEM) helps visualize cells, cytoplasm, and organelles. For this purpose, ultra-thin sections are required. TEM interprets atomic rather than molecular properties of the tissues and gives a two-dimensional image of the tissue. Scanning electron microscopy (SEM). SEM helps in the study of cell surface. Transmission electron microscopy (TEM) क्या करता है? Helps visualize cells, cytoplasm, and organelles. For this purpose, ultra-thin sections are required. TEM interprets atomic rather than molecular properties of the tissues and gives two-dimensional image of the tissue. Scanning electron microscopy (SEM) में क्या होता है? तो SEM helps in the study of cell surface. In this three-dimensional image is produced. The image is produced on cathode ray oscillograph, which can also be amplified. SEM can also be used for fluorescent antibody technique. Recent advances in microscopy. In the recent time, computers and clip technology have helped in developing following advances in microscopy: image analyzer and morphometry. In this technique, microscopes are attached to video monitor and computer with dedicated software systems. Microscopic images are converted into digital images, and various cellular parameters, example nuclear area, cell size, etc., can be measured. This quantitative measurement introduces objectivity to microscopic analysis. Telepathology virtual microscope. The examination of slides under microscope set up at a distance. This can be done by using a remote control device to move the stage of the microscope or change the microscope field or magnification, called as robotic telepathology. Alternatively, and more commonly, it can be used by scanning the image and using the high-speed internet server to transmit the image to another station, termed as static telepathology. Telepathology is employed for consultation of for another expert opinion or for primary examination. ओके फ्रेंड्स, ये चैप्टर यहाँ कंप्लीट होता है. नेक्स्ट चैप्टर है हिस्टोपैथोलॉजी टेक्निक्स एंड रूटीन स्टेनिंग. इसे हम नेक्स्ट वीडियो में कवर करेंगे. थैंक यू, बाय बाय. मिलते हैं नेक्स्ट वीडियो में. बाय.